In the last few videos, we've talked about different ways to move energy around a cell and to capture energy as it's released and to move it to a new location and use that energy to accomplish something. The two main processes we've talked about are the use of ATP in endergonic and exergonic reactions and the use of redox reaction, oxidation and reduction, to move electrons and hydrogens around because electrons contain energy. The reason we've been going over both of these is because we're now going to dive into one of the major energetic processes in the body, cellular respiration. Cellular respiration is a complex chemical reaction that can be summarized by the formula you see on the screen. So let's look at our reactants and our products for this reaction. The first one we have, our first reactant, it's C6H12O6. This is glucose. And seeing OS at the end should tell you immediately that this is a sugar molecule. It is possible to do cellular respiration with other molecules besides glucose, but this is the standard formula, so this is what we're going to focus on for now. So we start off with a glucose molecule, and we also need oxygen. So 6 oxygen molecules. These are our reactants. These are what's required. A series of chemical reactions, which we will go into in a lot of detail coming up very soon, will then occur. And at the end, we have 3 products. We have carbon dioxide and water both given off. These are actually waste products. We do not need them in cellular respiration. Carbon dioxide is given off in our breath because we have no use for it. Water, we do have other uses for. So while it's a waste product, we go ahead and often we'll keep that in our body and use it. But if you want a verification that we produce water doing cell respiration, walk up to a mirror sometime and breathe on it. Condensation will form. That is from the water vapor in your breath, which is caused by cellular respiration. The final thing we have is ATP. ATP molecules are energy storage molecules, and we produce, the number varies in estimates, but somewhere between 32 and 38 ATP molecules from a single glucose. You will never be asked on the AP test exactly how many are produced overall because there's some debate, but that's the ballpark to give you an idea. So what is the purpose of cellular respiration? Great, I've got a chemical reaction. What's the purpose? It's not to produce carbon dioxide, not to produce water, not to use oxygen. The purpose is to transform energy. Glucose and other food molecules contain high amounts of energy captured in their chemical bonds. We need energy in order to survive, to power an enormous number of chemical reactions happening in our bodies all the time. For example, I'm moving my arms right now. I am using energy to contract my muscles. The form of energy my cells are able to use is ATP. So I need to produce ATP. I need to take the energy from a glucose molecule, extract it from those chemical bonds, and then capture it in the form of ATP to be able to be used and powered other reactions in my body. And that is the purpose of cellular respiration. Before we go any further, I want to clarify something. When people use the term respiration, they sometimes mix it up with the term breathing. As a biologist, we need to be very particular about our terms. Breathing is the act of moving air into and out of my lungs. Okay, it's the physical act of moving the air. Respiration, or more accurately, cellular respiration, on the other hand, is the act of using oxygen for a series of chemical reactions inside our cells to transform chemical energy in glucose into high energy bonds in ATP. So let's take a look and see how this is going to happen. What actually is occurring in this cellular respiration step where this arrow is? Cellular respiration can be broken into a series of steps. The first step is called glycolysis. Glycolysis occurs in the cytoplasm of the cell, so it does not occur inside an organelle. It occurs in the cytoplasm. The second and third step, pyruvic oxidation and the citric acid cycle, are often joined together as a single step and are sometimes called the Krebs cycle, named after the scientist who discovered it. In your textbook, these two are separated, pyruvic oxidation and the citric acid cycle. I frequently lump them together So you'll occasionally hear me just call them the Krebs cycle and lump them together, but we'll make sure to go through each of them in turn. These occur inside the mitochondria in what is called the mitochondrial matrix, and we'll discuss later where that is and how it works. The final one is the electron transport chain, or ATP synthesis. This is our fourth section, and this occurs on the inner mitochondrial membrane. So again, we're inside the mitochondria, and it's in a different area than the matrix, and we'll go into the details of that. So we need to look through each of these in turn. What I'm going to do is I'm going to talk about glycolysis in this video, and then the others I'm going to save either for a later video or for actual class time. So let's start off with glycolysis. Here's a representation of glycolysis. Depending on the book you look at, you're going to see many, many different representations. So here's what I want you to do right now. In just a moment, I want you to pause the video. And I want you to see if you can figure out what the heck is going on here. Okay? So go ahead, pause the video, take as long as you need, and as soon as you either understand it or think you understand it or are totally mixed up, unpause and we'll walk through it. So go ahead and pause now. Okay, how'd you do? Do you get it? Glycolysis, we're covered. We can just move on to the next step. Odds are pretty good that you might have figured out some things here, but this is a little confusing until you understand some key pieces. So what I would like to do to begin with, talking about glycolysis, we're going to break this down into three parts. To begin with, we are going to start off with the center section. We are going to completely ignore what's happening over here and what's happening over here. So ignore those two sides. Let's focus on the middle. When you look at this, you probably figured out that we have a glucose molecule. But what do these black circles represent? In this diagram focuses on following the carbons of the molecule. Glucose is, if you remember, C6H12O6. It contains 6 carbon molecules. 1, 2, 3, 4, 5, 6. There they are. Those are our 6 carbon molecules. It is understood that this molecule also contains 12 hydrogens and 6 oxygens. But in this diagram, they're not drawing them. They're not important for what we're discussing. We are just going to track the carbons as they move along. So this is our glucose molecule, and we can see it has 6 carbons. The purple arrow represents a chemical reaction. Actually, since this is a highly simplified version of glycolysis, which contains a total of 10 steps, this contains several chemical reactions. We don't need the details of those chemical reactions. Don't worry about them. Just know that in glycolysis, the first set of chemical reactions are going to break our glucose molecule in half. So we're going to end up with 1 molecule and 2 molecules. These are not connected to each other in any way. So I could have drawn it differently. I could have gone like this. But to save space, they drew them one above the other. But understand, these are 2 separate molecules, each containing 3 carbons, so half of our glucose. This actually makes sense if you break down the name, glycolysis. Okay? Glycolysis, glyco refers to glucose, sugar. Lysis is the breaking of. We are breaking a sugar molecule. Glycolysis, glycolysis. So great, we're halfway done. We've broken our glucose molecule into 2 3-carbon molecules. And now another chemical reaction is going to happen. And each of these molecules is going to change into something called pyruvic acid. Okay, also called pyruvate. They are the same thing. Pyruvate and pyruvic acid, interchangeable terms. You'll hear me use both of them. Now, you might be looking at this and say, wait a minute, there's 3 carbons here and there's 3 carbons here. So how did a chemical reaction happen? Well, remember, we also have hydrogens and oxygens on this molecule. And so it is a possibility that we have gained or lost hydrogen and oxygens without gaining or losing carbons. So it's still, the new molecule, pyruvic acid, is still a 3-carbon molecule, but it is not the same 3-carbon molecule we had up here. You'll note this diagram does not give us the name of this 3-carbon molecule? We don't care about it. You do not need to know the name of every single molecule in here. It names the important ones. So, going through this again, we have glucose, a 6-carbon molecule. A chemical reaction or series of chemical reactions that happen, breaking glucose in half. We now have 2 3-carbon molecules. Another series of chemical reactions happen, converting each of these 3-carbon molecules into pyruvic acid. So I have 2 3-carbon pyruvic acid molecules that are going to go on to the Krebs cycle, the next stage in cellular respiration. That's the center section. So let me clear the screen and let's go ahead and talk about what happens on the sides. Okay, this time we're going to focus on what's happening on the right-hand side here. So still continuing to ignore this. This side you can see we've got some ATP and ADP molecules. If you remember in our previous discussions, ATP and ADP are basically a rechargeable battery. ADP is the uncharged version. Okay, here's where it gets charged. So it captures energy from the surrounding environment, charged up, becoming ATP. And ATP can then release energy and go back to ADP and a phosphate. And we do this over and over and over. So ATP molecules can provide energy to the cell, and ADP molecules can capture energy that is released to be stored and used later on. So what's going on here? Well, remember our glucose at first, we have a 6-carbon molecule that's going to get broken into 2 3-carbon molecules. Glucose is a pretty stable molecule. That breaking doesn't happen naturally. It doesn't want to occur. It requires input of energy. Have you ever heard the phrase, you have to spend money to make money? Well, here, you have to spend energy to get energy. So what is going on is breaking of glucose requires an input of energy. It is going to require 2 ATP molecules to provide the energy needed to break our glucose. When that happens, the ATP molecules are going to be broken down into ADP. That 3rd phosphate is broken, that high energy bond, releasing energy. That energy is given off and captured in these chemical reactions to break the glucose molecule. And our ATP becomes ADP. So we just spent 2 ATP molecules. So we lost 2 ATP. 2 ATP fewer in the cell than I had when I started. So now we have our 2 3-carbon molecules. And another chemical reaction happens. However, this chemical reaction is exergonic. It gives off energy. So the formation of pyruvic acid is going to give off. Bonds are being broken and energy is being released into the system. And enough energy is being released that we can take 4 ADP molecules that are floating around and they can be charged up into 4 ATP molecules. So we can gain 4 ATP molecules. So we had to spend some ATP to break the glucose, but then the next reaction releases energy so we can form ATP. So one of the questions that comes to mind then is, how much ATP do we generate from doing glycolysis with 1 glucose molecule occurring, going through this process? And the answer would be, we have a net gain of 2 ATP. Now you might look and say, wait a minute, we made 4. That's true, but we spent 2 to get there. So even though we gained 4, we lost 2. So we end up with 2 more than we started with. So we would call that a net gain of 2 ATP. And that's what's happening in the right-hand column. So let me erase it again and let's take a look at the left-hand column. So now we understand what's happening with the glucose. We understand what's happening with ATP. Now we're going to go ahead and we're going to look at what's occurring in the left-hand side. Here we have 2 molecules we talked about in a previous video. NAD plus and NADH. And if you remember, these are for oxidation reduction. NAD plus gets reduced and becomes NADH. NADH gets oxidized and becomes NAD plus. When it is reduced, it gains hydrogens and electrons. When it's oxidized, it loses hydrogens and electrons. Now, we can't see the electrons, but we can see the hydrogens, and so that's what we follow. So what's actually going on over here? Well, in this column, nothing's happening up at the top, so this reaction doesn't have any oxidation reduction. But when we go from these 2 3-carbon molecules, again, unnamed, don't care about the names, when those are turned into pyruvic acid, not only is energy released for ATP to be formed, but a reduction reaction happens. This molecule, whatever it is oxidized. Okay, it's losing electrons and hydrogens. Where are they going? Well, the NAD plus is grabbing them, and it's grabbing the NADH. The H is being added, also electrons are being added to that. So NAD plus is reduced, becoming NADH. It grabbed the electrons and hydrogens that were released in this chemical reaction. There were 2 molecules here, both of them reduced in NAD plus to NADH, and that's why we have 2 here. So what happens to our NADH? The NADH is going to go down and it's going to skip the Krebs cycle and go straight to the electron transport chain, the final step of cellular respiration. So we'll see that later. That's going to be important in the last step. But remember, we're generating energy in 2 different ways here. The first way, we are directly stripping energy off of this molecule and using it to form ATP molecules. ATP is the form of energy we want. We can use that later. That's great. These ATPs are ready to go. But we are also doing a series of oxidation reduction reactions, and we are reducing NAD plus to NADH, stripping small amounts of energy off this glucose molecule. And we're going to use that later to create more ATP. And you'll see how that works coming up. That's glycolysis. So what I encourage you to do in just a moment when the video's over is try to explain what's going on here again. See if you can walk through what's going on. I'm going to stop at this point. We do have 2 more parts. The Krebs cycle, which oxidative phosphorylation, citric acid cycle combined, and then we also have the electron transport chain. But I'm going to talk about those in class or in a future video. You've got enough to work with right now, so think about how this goes together and be ready to look at the next steps next time. Okay? Good luck, work your way through it, and if you have any questions, make sure to ask them in class.